Yes, exactly. Thank you for that. I completely forgot. Right. Welcome everybody to the Oslo AI meetup in the collaboration with Neural Met, which is us. I will be your gracious host. My name is Dennis Larsen. I come from Oslo Met, from Neural Met. We are a student organization that's specializing in creating knowledge and getting students interested in artificial intelligence. And we create workshops and meetups and things like this for the students and other people. Today we will be presented with some of the current research into the cross section of AI and health. We have our first speaker, who is Jonathan Whitlock. He's an associate professor at NTNU, and he is the head of the Whitlock Research Group at the Kavli Institute of System Neuroscience. Our second speaker is Pedro Lin, who is a professor at Oslo Met with me. The third speaker will be Vashira Tamwati. I hope I spelled that or I pronounced that right. Vashira is a postdoctoral fellow at Simula. And the fourth and last speaker will be Bastian Zapp, who is a research fellow at the University of Oslo and is part of the Psi machine learning project. Towards the end, we will have a brief discussion regarding the futures of AI and the health sector and its ethical implications with some questionnaires, if anybody would like. And then towards the end, for those interested, we can be in the networking in Gather Town. Right, so I am happy to present our first speaker, who is Jonathan Whitlock. And take it away, Jonathan.
Sure thing. Let me maximize this and see about sharing my screen. Great. Okay, so you can see my screen. All right, you see, we good? We sharing?
Yep, everything's perfect.
Okay, so the perspective that I'll bring from my talk today, I'll minimize that so you can see my slide, is that of a neurobiologist. We use rats and mice here at the Kavli Institute to study how the brain produces behavior. What I'll be talking about is the general goal of systems neuroscience and understanding behavior in the brain, and how these are really, as with any complex, multi-part, high-dimensional system, these are eternal markets for machine learning and applied statistics. So I'll just get right into it. I will introduce the work of my lab kind of along the way. I'll just show it by example to try to stay on time here. So in behavioral neuroscience, the overarching goals are twofold. One is to understand animal behavior, if possible, naturally generated animal behavior without perturbing it. And at the same time, you want to understand systems level neural computations to understand how these things together, if you can understand how these things fit together, how behavior fits in one scale and complex patterns of neural activity work together on the other scale, then you put those together and you can start to better understand how the brain generates behavior. You can in turn understand how behavior and experience and social interactions shape the brain. And eventually you can generate better on-target treatments for neurological and psychiatric diseases, right? When you can, again, understand how the behavioral and neural elements fit together. So in the quest to understand behavior in the brain, there have been different traditions. So one of them that goes much further back in the 20th century is to use very strict, controlled, simplistic behavioral tasks. And that's epitomized by this picture here from the lab of Vernon Mountcastle. This is in the mid-1970s. And this is a very typical type of behavioral paradigm that lots of labs still use today, where the animal is head fixed, it's sitting in a chair. This is a stepping recording drive that's affixed to the animal's head. Even though the animal doesn't feel any of it, it's just an open craniotomy in the animal's head. You can record neurons while the animal is awake and behaving. It can respond to discrete sensory stimuli that it would see with its eyes, and then it can reach out, and push a button. And the advantage here is that, the behavior is controlled. You're removing extraneous aspects of behavior like fidgeting or things that you don't want the animal to do. You can get lots of repetitions. Right, so that's one approach. Another approach that's epitomized by the mouse type of work, and this is more like what we do here at the Copley Institute, is that the experiments are not really well controlled, and the animal's behavior can be much more complex. So here, instead of the animal being bolted into a chair, you use a teeny tiny micro drive on the animal's head, and the animal actually carries it around itself. So it's portable. And here, this is an example of what's called a flex drive. This was published a little more than 10 years ago. That the drive weighs 2 grams. The mouse, it's like 1/10 of the mouse's body weight. And that's great because it affords you flexibility, right? The animal can behave naturally. And here is an example that now in the video here, this is a rat. It's not a mouse. And this is a neuron. That popping in those red dots right there, that's from a single neuron in the animal's brain and a structure called, I'll pause it, a structure called the hippocampus. And we know that this structure represents space. It's important for spatial mapping, right? You can see it because the rat's free to move around and you can hear the cell firing. You can put the two together and say, aha, there's a place field. That's what they're called. Right, but the thing is, there's a single camera that's overhead, right? We don't really have a very good portrait of the animal's behavior. Is it like, what's the animal actually doing? We can just see where in the box is the animal. So the goal of my research program is to kind of make the best of both worlds here so that you have really highly structurally controlled measurement over the animal's behavior while the animal's freely behaving. So to that end, this is a paper that we published a few years ago. We tracked the animals in 3D. We marked the backs and heads of the animals and tracked them with a 6-camera infrared tracking system. And I should note that this was the PhD project of former student Bartol Mimica and my first postdoc, Ben Dunn. So when we put all of this together, our data sets now look like this. And you're probably all seeing this kind of thing for the first time, but in the field at large, this is quite new, right? So normally animals are just viewed as like a dot, moving on a surface, right? Where was the head of the animal? Whereas here we can see, the full complexity and 3D aspects of the animal's behavior. And so here's an example of one of the recording sessions. Here's the rat that's actually running around. The orange dots here are the markers that are on the animal's back. And that little popping sound you hear in the background, right there, that's a neuron that's discharging the animal's secondary motor cortex. Its frontal motor cortex is the animal's freely running around, right? And there's the rat. You see it's marked up. It tolerates the markers quite nicely, right? Freely behaving, but we actually have pretty good quantitative control over what the animals are, what their behavior is doing. Okay, so this is cool, but it creates a really, really complex high-dimensional data set with lots of moving features and lots of moving parts and different covariations that you can track between the back and the head and the neck of the animal. Okay, so we can take the 3D pose of the animal and we can measure discrete behavioral features like head azimuth, like the animal's head is turned left or right, or if the back is turned left or right. And as you saw, the animals are moving around, right? So these postural features are changing through time. So then if you imagine that each one of these postural features can be read out as like a squiggly line that goes from lowest to highest, right? Let's say as the head goes from left to right. And you take that different postural features are expressed at different time scales. What you do is what's called a Morelet wavelet decomposition. This is like a time frequency decomposition of the changes in posture through time. Then this creates a very high dimensional clustering of different postural states that coexist when the animal does different behaviors like grooming or rearing or whatnot. And then you take that and use basically linear then nonlinear low dimensional embedding. So this is like grazing in with neuroscience or grazing in with machine learning here, using t-SNE. So the idea is you take these clusters of behavioral features and you project them down onto a 2-dimensional surface with low dimensional embedding. And each one of these colored patches of dots here corresponds to the co-occurrence of postural features that happen during different behaviors. So you can do watershed clustering. And now what we have is a map here where each different color on this map tells us what behavior the animal was engaged in a certain point in time. Like, was it rearing? Was it turning? Was it drinking from a well? What was it doing? Right. And when you take the video, like I showed you a second ago, and what we can do now is just fast forward and zoom and look at specific behaviors whenever the animal was rearing up in the top right over here, or if the animal was running straight, right? The video slows down whenever the animal engages in that behavior. And so this is very, very powerful because now we have a freely behaving animal. We can track its postural kinematics in 3D, and we have temporal control, like the monkey in the chair doing the same thing again and again and again. But now the animal is willfully producing its own behavior. And so it's possible to study things much more quantitatively now in freely moving animals. You can ask things like, does it matter what task the animal was engaged in when a certain behavior was expressed? Does that affect the way that the brain was encoding it? You can ask, does it matter what behavior the animal did before, what it's going to do next? Does that change the way that the brain encodes a certain behavior? You can look at how, for example, how behavioral representations are affected during social interactions. It opens up lots of doors. That's the point here. And now, just to kind of like broaden it up a little bit beyond, you know, that example of my group's work, deep learning has already been applied to successfully model not only human movement, but movement in lots of species, right? So here is an example of a generative model that my former collaborator, Graham Taylor, had made during his PhD, did this with Jeff Hinton. And this is actually the output of a generative model that was just trained on a few hundred frames of a human moving, right? And here's another one where it's like they're taking it and making it do kind of like a sexy walk, right? And he was doing this using factored conditional restricted Boltzmann machines, FCRBMs, that could capture different aspects of the pose dynamics that people were generating, and it could add styles on top of it, right? And so the kinematics of the individual in this case are captured by a multi-layered restricted Boltzmann machine. So this is another way in which machine learning can be used and applied to model movement and behavior. Similar approaches have been used to document rats. This is a lab kind of like mine that wants to study freely behaving rodents. This is Bob Dada's lab. He's at Harvard. And what you're seeing here, if you look on the top left, these are the movement profiles of mice that have been tracked with an XFox Connect, right? So it's very simple, just a depth profile tracking system where you can record the holes of the animals and color code the height, right, in different colors. So as the animal rears up, they turn pink. But the point here is that he just used a two-layered autoregressive hidden Markov network to capture the 3D pose dynamics of the moving animals and captured the transition points using Markovian modeling. And to broaden it out even more, convolutional neural nets have also improved a lot in the last 10 years. And this has led to an explosion of markerless tracking tools. So here are examples of software called Deep Lab Cut that, again, it's a convolutional neural net that is trained on a few hundred frames where the user labels parts of interest that you want to study and track. And you can don't even have to put markers on the animal now. You can just let the animal move freely. So here is an animal. This is, I believe, is a Parkinson's model rat that is being used to study motor behaviors. The animal's reaching out to grab a pellet. You can. There's an example of a head-fixed animal that is running on a running wheel. Again, you don't have to mark anything. The neural nets are so good now that they can track these things themselves. Here's another example of work from my own lab where we're using facial tracking on rats to track the eyes and whiskers of animals, again, as they're moving freely. And we're also tracking them in 3D. You can also visualize collective behavioral dynamics. And this is from Ian Kuzen's lab down at the Max Planck Institute in Germany for animal behavior. Here, the 2D posture of the animals can be defined and tracked as the animals move freely, and you have a school of freely behaving fish. You can look at things like proximity, structure, dominance, again, all in the context of natural behavior. So this is just opening up lots and lots and lots of doors to study interactions between genes, brains, behavior, and even social behavior. And this is just a throwback back to, this was MoDeep, one of the earlier algorithms that came at the beginning of this recent convolutional neural net craze, in which it was demonstrated on videos with lots and lots of people that you could do markerless pose tracking there as well. Okay, so again, the overarching goals of behavioral neuroscience, we want to understand natural behavior, and with advances in tracking and machine vision and behavioral modeling, there is just an explosive phase of advance that's going on right now. So it's a very exciting time to be doing behavioral neuroscience. And I didn't have time to talk about any of this, but there's also neural engineering and recording advances and modeling approaches of neuronal population activity that's also leading to this really kind of a revolution that's going on right now in terms of understanding population coding and interactions between networks of neurons. And so these things can be put together now using things like dimensionality reduction, statistical modeling with deep neural nets or artificial neural nets. And again, this is definitely bringing us closer to our goal of understanding how the brain generates behavior, and eventually brings us closer to the goal of generating drugs and understanding diseases and trying to prevent disease. So I'm not sure if I went over time, but thank you for your attention. I have to thank the members of my lab who made my work possible, and my funding agencies and the NTNU. So thanks for your attention.
All right, thank you so much, Jonathan. That was extremely interesting. Then, while I was listening, you were getting all of the buttons that I like. There's a lot of fascinating stuff there. Cool. Right, we're actually a little bit short on time, so we're going to jump right next to our second speaker, whose name is Pedro Lin, who is a professor at my university, Oslo Met. Hello, Pedro, are you with us?
Hi, Dennis, can you hear me?
Yes, we can hear you perfectly. Are you ready? Do you want to take it away?
I am now sharing my screen.
Yes, we can see it perfectly.
Can you confirm? Very good.
All right.
Thanks a lot. Great. It was a great presentation. I like very much Jonathan's presentation. I will not present any results today. I decided basically to focus and discuss a little bit more ideas and focus in two of the projects that we started now in Oslo Metropolitan University. So before going to that, I just want to perhaps to make a step back and put it a little bit in perspective what I was trying to think when Dennis invited me to speak in this AI in Health event. So I think it would be, I just saw here, okay. Can you see my presentation? Because I had a notification that, yeah.
We see AI in health. Wow.
Very good. So, and why that? So basically, I would say that there are two important things. That one is our society, where probably we want a better environment for everybody in this context, in the context of health. And the other is about the future, and the future not only about AI itself, so how to develop, but also how to put it sustainable. So in what concerns society, I think AI plays a fundamental role because it helped us in many things, but it's also important to understand how people deal with these tools. So trust is something that is also important, and because of these two dimensions, sustainability and trust, we started this year at Oslo Met a center where we try to focus on different aspects related with these two approaches: how to make AI sustainable, how to make it trustable. I can perhaps, if you want, then speak a little bit more about this initiative. We have here different research groups within the center. I will basically today focus more on these two, quantum computing, and those then related with medical data. But if you want, then during the discussion, I can also tell a little bit more about the others. So the first project I would like to speak about is a project that is being coordinated by Oslo University Hospital, here also in Oslo. But it's a huge European project. It started this year. It involves 8 countries with 15 different partners. So I invite also you, if you want then to see more about it, the activities that we are now unfolding in this website. The main idea behind this project, AI Mind, is to approach a huge problem, which is dementia. So nowadays we know in developed countries at least people live longer, that's true, but also because of that, the frequency of diseases related with dementia is also increasing. Now, what happens with this kind of diseases is that if they are detected, diagnosed early enough, then it's possible to act on it. There are already some pharmaceuticals that could act on it to try to prevent it, or at least to slow down its development. But of course, what is really difficult is really to detect dementia in people at very early stages. So the aim of AI Mind is precisely to try to find ways from data from patients in order that we can have algorithms that can, with up to a certain accuracy, try to detect at least the risk that a certain person is possibly developing dementia. Now, as I told you, we have these are 8 partners. It's the project is being coordinated by Oslo University Hospital, and the role of OsloMet is to work in 2 of the work packages together with the partner in Finland around these 2, so to say, big components of the tool that we are aiming to develop in AI Mind. So one we call connector and one we call the predictor. So the connector basically will try to map the set of different signals that we can take from the brain. Typically, there will be surface EEG signals, and try to map this into a certain functional connectivity brain network, so-called. What is this? Is basically a network where each signal is represented by a node, and then it's linked with a certain strength, with a certain weight to the other channels. And the weight of each link is measured by a certain metric that tries to account for how strongly related these 2 channels are. They can be so simple metrics like covariance matrices, but they are also more sophisticated metrics in the scope of statistical tools like mutual information and entropy-based metrics between these 2 signals. Now, what we can also do is that instead of having this statistical approach to try to derive a functional connectivity network from the multi-set of data, we can also apply some of the tools that Jonathan was also mentioning. So neural networks or graph neural networks in this context will also be tools that we will also apply here. And the main idea for this component of our tool is basically then to have a kind of map of the brain for this person that should have a certain topological pattern. So the weights can be then represented, and we can now try to relate this topology, this structure, with other kinds of data sets, particularly from genomics, in order that, and then train a model to see if we can map both together with a known diagnosis of dementia in these patients. So I don't know who is the partner now taking care of data collection, but we are now trying to launch a campaign to collect data from people that volunteer basically to in the next years to provide this data from themselves, and then try. They will have a clinical follow-up in order to see which of these people will then develop dementia, and then we can have proper data to train our tool. I apologize, this figure is not with sufficient resolution, but of course that having this connector, so you see this is basically the functional brain connectivity network that I was talking about. We can then bridge this with the genomic data and then train also this kind of tools from machine learning. This, I think, it's a tensor factorization tool that will end up then trying to make a prediction, or at least to make an estimate about the risk level that the patient should have to develop dementia in the near future. So this project started this year, and Oslo Met team is basically me and two other colleagues, and we will have now starting in one PhD started now, and one second should start next year. The second example deals with cancer treatment. So as you may know, cancer has many ways to be attacked. So there are today also lots of different therapies to try to treat cancer in specific patients. And the big problem is basically what is the best therapy for a certain patient. It is known that there are therapies that work wonderfully with certain patients, but not at all with others. And so there is today, it's a field developed within mathematical modeling, where people try to develop models that they need to be parameterized to assess, to try to explore all possible combination of different therapies for cancer treatment, and in the end try to derive the one that is optimally for the specific patient based on the clinical data that we have from that person. Now, of course, this is a and what we do basically is, when we use then a classical computer, basically we need to explore all the possible options, and this ends up, as you know, with a kind of with a problem that is in practice not solvable, in the sense that if we increase the amount of data, or if we increase the amount of combinations, we will end up needing a huge amount of time to have an optimal solution, which is, for practical purposes in the clinical context, not feasible, so to say. So the alternative using computers is then basically making parallelization. So we will try now to have not one computer, but lots of them, each one dealing with a small set of different combinations, explore all of them in parallel, and then we can try to see what is the best combination possible. So here we gain in time, but of course we will lose then in the computational cost in the end. Now, this is exactly here the point where we come with the motivation from what is now emerging technology, which is quantum computing. So a quantum computer is basically a computer that has not these binary bits 0 and 1, but due to quantum effects, so it's a completely different technology, can have a bit that can sweep between all the values between 0 and 1. And basically here what we see is that if you can basically implement an algorithm that could explore or can implement the mathematical modeling for cancer therapies. Basically, what we are doing here is that we would then have a quantum algorithm that combines all possible combinations into one single task, so to say, and so can in one step or in a few steps do the same with one single quantum computer as many classical computers do in parallel. So in this scope, I would like to say that last week we got we just launched the first quantum computer in Norway that we bought recently. It's a very small one. It will not solve anything in cancer therapy, of course. It has only 2 qubits, but it's already a starting point to involve and to teach our students how to implement quantum computing algorithms, which is also from the point of view of programming, it's a completely different approach. Now, specifically for the cancer therapies, what we are aiming now to do is to implement something that is similar to simulated annealing. So for those that do not know, so in optimizations, we have basically a very complicated surface, and that represents the function that we want to optimize. And optimize means basically to find the minimum value here, and this can be done through what is called a simulated annealing. What simulated annealing does basically is that if we visit this region of our model, of our parameters that we have here, a local minima, we will then enable to make a jump with a probability that that decreases, so to say, with the height of these hills, and then find another minimum till it converges to the global minima here. So now the problem here, this is a classical approach that is easy to implement, but the problem here is that sometimes we have a huge height that could be very, very high, and so this would make the probability to make this jump very small. Now, in quantum, what is called quantum annealing is a different way that we will not consider not only this height here, but also the width of this hill. If the width is small enough, so to say, we will have a reasonable probability. Whatever it doesn't matter how high this hill will be, that we can through annealing go from one minimum to the other. So simulated annealing already exists for cancer therapy. This problem is already implemented by our colleagues in UIO. Now, the idea is basically now to map this into a quantum annealing into the quantum context, trying to come out with a quantum annealing algorithm for it. And this is basically the two ideas that I would like to share with you, the 2 projects. Thanks.
Thank you so much. Pedro, that was extremely fascinating. I myself actually just did a course on quantum computing, and the quantum annealing processes are extremely fascinating, just in a way of training neural networks and kind of using the intrinsics of quantum mechanics into trying to teach a neural network with quantum tunneling and the superposition of qubits. That's extremely fascinating. Let's see, our 3rd speaker is Vajira, but unfortunately he is experiencing some internet troubles right now. So I will actually go ahead and play his presentation for him. Let's see, and I do believe that if there's any questions to him, that you can freely ask in the chat and he might answer anything that people are wondering. Let's see if we'll be able to share this thing. There we go. And if people can confirm they can see my presentation. Good. And then,
Hello everyone. In this talk, I'm going to discuss about AI in medicine and magic computer from environment and what I suppose.
I think you might might need to have the sound on.
Correct data, then we
Is
have
it muted?
Experts knowledge.
Yeah, now
and
it
correct
works.
data in
Okay.
medical domain. Finally, we can train machine learning methods that it can models using this data. At the last stage, we should produce final products. In this final product stage, if you are interested, we can practice experimental artificial intelligence methods. However, we realized that while we have more papers about applied machine learning methods in medical domain, we cannot see final products in real world. When we were searching about reasons, we could identify that this data is the biggest problem in medical domain compared to the other domain, because in medical domain we face privacy issues, and also we face this annotation problem, because in medical domain we have to use expert knowledge and time to enter data. While keeping those four stages in mind, I'm going to discuss three different data sets using examples. First one is Ecg data set, which represents time series data, and the second one is GI-track data, which represents images, and the last data set is sperm data set, which has videos of sperm samples. Using these three data sets, I'm going to explain different type of AI application in medicine. First one is the Ecg data. Using this Ecg data, we were interested to predict PR interval, QT interval, J evolution, that kind of properties of ECGs using different techniques. As a solution, we have tested residual connection-based deep learning model.
right, we're just going to pause a little bit. I do believe that people are struggling with the sound. Is it a little low? Yes, let's try to do that one more time with maybe a little bit.
Dennis, it may be that you are selecting the wrong micro.
Yeah, exactly. It's on the different microphone. Let's see. If we just choose a filter.
There's no sound.
Let's see, bear in mind, we're experiencing a little bit of difficulties. Let me just try to do it in a different browser.
Maybe try to download it and play it from like a media player and share just the media player itself.
That was exactly my idea. So I'm just trying to download this real quick. There we go. How's everybody doing so far? Everybody's okay? Good? There we go. Share. Let's see. What if we do? Sound? Anybody hearing the sound?
No sound.
That is extremely fascinating. Zoom is a wondrous program that doesn't really want to do what you want to do. I'm wondering it's because we're recording and I am not, but then you had a sound.
Maybe should we jump to the next?
Yes, I do believe we are actually need to do that. I will talk to Vashira, and if it is okay, then we will share the video for those that are interesting. I do apologize for the inconvenience. For some reason, this is not working at the moment. But our last and fourth speaker is Bastian Zapp, who is a research fellow at the University of Oslo and is part of the Sci machine learning project. And I see that Bastian already started his presentation. And then if you're ready, then take it away, Bastian.
Yeah, thanks, Dennis. So my name is Bastian and I'm actually a PhD student here at UEO. And I'm in the SciML project, which comes from scientific machine learning. And what we basically do there is combining differential equations with the machine learning algorithm. And today I want to show you a bit of the work that I've been doing in using these tools to model fluid dynamics in the human brain. So I will divide this talk into 3 parts. I want to convince you why we do it, that it's interesting, how we do it, and what we have found so far. So the reason why we want to study the fluid dynamics in the brain is that in the brain, we don't have a conventional lymphatic clearance system. So in the rest of the body, the lymphatic system takes care of excess fluids and waste product and so on. But as you can see in this figure, we don't have that in the brain. So the question then is, how are waste products and excess fluids and so on cleared from the brain? And that's especially important with respect to dementia and so on. So for example, in Alzheimer's disease, what one sees is that a specific type of proteins builds up in the brain, and that's these brown things you can see here in this image between the neurons. So the question is, how does a healthy brain clear these waste products? And what has been suggested like about 10 years ago, what was that it's actually the cerebrospinal fluid, which is some sort of water surrounding the brain, is clearing the brain from waste products. And the way it is believed to work is like that. So you have the water around the brain, and then it's entering the brain along arteries, and then it's being somehow transported between arteries and veins. And then on the way from the artery to the vein, it takes out waste products. And that's kind of the medical theory that we want to investigate. So the question then is, what kind of physical laws does the flow in the brain satisfy? Is it just pure diffusion, or is it also convection? And that's kind of the hypothesis here, that it's convected flow between the arteries and the veins transporting out the waste. And the way we can study this system is MRI tracers. So the experiments that have been conducted were like the following. So take a scan to get like a baseline image of the patient, then they inject the tracer into the cerebrospinal fluid, and then you can observe in these images how the tracer spreads throughout the brain. Then, in light of the hypothesis, we can ask, what kind of model explains this data? And the simplest model we could have is just diffusion, no convection, and also constant diffusion coefficient, which then makes this a really simple equation. This equation we can model on the, can simulate on the human brain and then find the diffusion coefficient that best matches the observations. And that has been done using traditional numerical methods. And what was found, previous work in our group, was that the diffusion coefficient that really describes this data quite well is a bit larger than what has been measured otherwise. And that would then, of course, support the theory, because if there is convective transport, it would be faster than diffusion. But the problem with this approach is that we kind of have to iterate between solving the PDE and minimization of the cost functional in every iteration. So these computations take like a couple of days on a supercomputer because of this approach. What we could alternatively do is to combine the constraint into the cost function as a sort of a penalty, and then instead of iterating, only minimizing this cost functional. The question then, what kind of function do we use to model what we see in the images? And it can be a neural network, and that's what we do here. So the function C that's modeling the data is the output of a vanilla feed-forward neural network, which takes space and time as input and predicts the concentration. The way we can visualize is this. So we have the time and space as input coordinates, and we have some nonlinear layers and so on. Put out the concentration C. Now we can use automatic differentiation to compute all the gradients and so on, and assemble our PDE residual here. Combine these two into a loss, minimize that, and hope then that by minimizing this loss function, we fulfill both the PDE and match the data. And this additional term gives these networks the name physics-informed neural networks. Have been proposed a couple of years ago and have been very popular, have become very popular in scientific computing since then. What we now do is we test that on the data we have from the hospital. We take a subregion of the white matter and have as data these observations at 5 time points with the tracer, and we can then try to minimize this cost functional. And then if we compare the data, that's what you see here in color. So red is like a lot of tracer, blue is basically no tracer. Up here and down here, that's the prediction. And it's the prediction kind of overfits the data, we would say, because there's a lot of noise in this observation. So there's quite some high error here. And in addition, we get an unphysical estimate for the diffusion coefficient. So the question is, what's wrong with this approach here? What we did, we created some synthetic data by simulating the diffusion equation on a synthetic on a brain mesh. And the data then looks kind of like this. We can compare that to the data from patients, and it looks quite similar. So it's a reasonable test case. What we then see is that we still have some quite high error between the blue and the green line, because now we have ground truth. We know what kind of diffusion coefficient it should yield, and it doesn't. The question is then why? And it turns out that if you smoothen the brain, so if you have less complex geometries, it works better. So here we have less error, and then if we have this potato brain, it's almost perfect. So what can we do about it? And it turns out that there's a simple trick we can do, and that's about the formulation of our loss function. And usually people would use the L2 norm here, but it turns out that in these cases, it helps to switch to the L1 norm. Still works perfect in the simple case, but now we're actually to solve the complex case as well. So that's what you see here. The red line almost touches the green. Then we can do that on the real data as well. And now we can see that this trick actually helps us to get these more reasonable predictions and also diffusion coefficient, which is not vanishing anymore, which is somehow close to what we found before. So we are getting somewhere. And as a reference, that was like this first case here, where we had this overfitting and the vanishing diffusion coefficient. So that's what I wanted to show, and maybe to give an outlook where this method could be used in the future is in other problems related to CSF in the brain. And what has been reported is that there's a difference between sleep and awake, and that's what you see here. So if people sleep and don't sleep, there's a big difference how the tracer distributes in the brain, and maybe these methods could be used to infer why or where the difference comes from. And also there's difference between patients, and maybe we can find some patient-specific parameters and so on. And that's what I wanted to show today.
Thank you so much, Bastian. That was fascinating. Just a question, actually, I was wondering something. So is this the brainwash that we have recently learned, that every time we sleep, there is a wash fluid that happens in our brain that kind of takes away all the stuff? Is that what you were talking about?
I think that's probably
yeah, the same thing. That was very fascinating, because every night we actually get brainwashed by our own spinal fluid and get cleaned out. I've been reading about that a little bit. That's extremely fascinating.
Yeah, and one thing to keep it. Yeah, sorry.
No, go ahead.
One thing to keep in mind is that for these models, they use rat experiments and so on. So it's quite different than the human brain. So all these theories, and that's what you said with this brainwash and so on, is probably based on mice experiments. And here we have this opportunity to kind of look and compare what's happening in humans. So
feels like the AI will bring in a new era of analysis and brain analysis and pathways, and it's extremely fascinating. I think Bastian was, and I know for a fact that he was the last speaker. And right now we'll have a little discussion. So if there's any questions or anything that people are wondering for any of the speakers, or anything at all, we are opening the floor. Either chat or just raise your hand. I do believe that everybody has the ability to unmute. Yes, Pedro.
Thank you, Dennis. Perhaps to Bastian, very interesting results, Bastian. I was wondering, is there any reason why you don't use the channel formulation for the flow with convection? Because basically it's just one term and you can always solve it if you have that in your integral. I cannot see now any reason for not doing it, since you are just solving it numerically.
So did I understand correctly that you ask why we exclude the convection term?
Yes.
That's basically because if you think of this model, it proposes that the convection happens between arteries and veins, and they are so close together that we cannot resolve that on the MRI. The question would then be,
okay, for a physical reasoning. The other thing is that if you would assume there is this diffusion in complex geometries, in this porous media and so on, which is basically this fractional diffusion, I think it's the term, and I don't have now by heart the equation, but it mimics pretty well this exponent p that you are introducing there. Have you checked that with fractional diffusion equation? Because it could be a good alternative, since you are trying to see how to improve further your results.
Yeah. So actually these diffusion coefficients that we compare with, they take into account these complexities and so on. So it's the apparent diffusion coefficient we compare.
That was interesting. I did not understand any of that, but it sounds fascinating. Bastian, would you be able to explain very in layman's terms what it is that you guys are talking about?
So I think what it boils down to is that if you have this complex geometries and porous media and so on, the diffusion will actually look different than in a bulk medium where you don't have any pores and so on. So even though the material might be the same, if you have pores and so on, the diffusion is slower.
Oh yeah.
So if you look at it from the outside, it appears different, and that's why people talk about the apparent diffusion coefficient, and that's the one we are comparing here, which kind of takes into account these pores and so on, because the brain, is a porous medium. Does that make sense?
Kind of, actually, that makes sense. Like when you think about it, like through the pores, it does.
Can I add one thing? What Bastian is saying. So
go ahead.
It involves a concept which is called fractional derivative. So a derivative, everybody knows, if you have kind of the position in the brain, then the first derivative of the position would be the velocity of your flow. The second derivative would be the acceleration, and then you could explain the dynamics on it. Now, what happens is that this would be true in this very degenerate case that Bastian showed, when the brain is basically it's a kind of a continuous space where the flow is free, so to say. Now, that doesn't happen. There is a complex geometry where that enables the flow only to visit some parts. And this can be now modeled, the dynamics. It's no longer only using first and second derivative, so an integer derivative of your property, but a fractional derivative, which is it has a mathematical definition. It generalizes the definition of derivative, and it enables them to formulate evolution equations in these complex geometry cases.
Wow. Yeah.
It's the best I can without maths.
Well, and maths would just be even more difficult. We have a question here for Jonathan. Jonathan, are you with us?
Yes.
Yes. Have you experienced a lot with different approaches and algorithms for your clustering data? You showed that you think that it's PCA, TSNA for the moment. Is this a source of future work, or does it work satisfactorily well?
Yeah, it depends on how you define satisfactory. You know, I mean, people develop these methods and they take years to optimize them, but really in the end, it's just about passing an eyeball test where you as a human with your own, you can think of the human eye and brain as a really well-trained, visual object recognition algorithm. So what you can do with the current approach that we use is the utility of it is that it generates timestamps, right? So you can have an animal doing a structured task or an unstructured task, doesn't matter. But what you can do, the critical thing is you can load up a video of the animal performing, and you can fast forward to the points where the algorithm thinks the animal's doing a certain behavior or not. And you can see how consistent it is. Is it error prone? Is something being mislabeled, or is it very inconsistent? Is it, how error prone is it? And then it's kind of back to the drawing board for reparameterizing it. I think conceptually, so I'm still kind of blown away. So the pipeline that we use is from, I guess he does the physics of biology. His name's Gordon Berman. Came up with it when he was a postdoc at Princeton. And it was based on segmenting behavior in fruit flies. And the concept of it is so, I don't think it can get simpler than this. You decompose the video, the animal that you're tracking, into like sticks. You make it the simplest possible geometric form it can have that has temporal dynamics. And then all you're doing is just extracting very, very basic temporal features or covariations between features that you're tracking. And doing PCA basically takes those features and shoots them out into component space as far as that they can go, right, orthogonally. And then you just throw this down in 2 dimensions, right, to visualize it. This, I mean, I don't think it gets simpler than that. I don't know if it can. There are lots of different machine vision algorithms that are out there now. And it's sort of like walking down the street and everyone, like a bunch of taco stands or street food, and everyone's like shouting out that their stuff is the best. And so what you have to do at the end of the day is really just take what passes your eyeball test. What system do you have that's optimized, that works well for you? When you see it, you give it a sanity check, you know it works. So we actually tried the ARHMM, this thing that was developed by this guy at Harvard, that was originally developed for human pose tracking. And as it turns out, for our data, for our tracking data, it was, I want to say useless, but we could just never get it to work because they were using the hulls of mice using the Xbox Kinects, which has its own, temporal dynamics. That our data, which is kind of like a stick figure where you have the back and head of the rat, that our model didn't have. So what works really, really well in one lab is not necessarily the magic sauce that's going to work for everyone's lab. So it's like what you develop and what you get working best in your hands is what you stick with. And if you developed it yourself, then you scream and shout and write review articles and say how yours is the best. But I don't have a dog in the fight. Like I'm just a consumer. So I just take what works best, and we happen to have this tool that happens to be working really well. But there are many tools that we could use. I hope that answers it.
Thanks, Jonathan. I appreciate it.
Sure thing.
You have a fairly complex pipeline and a complex stack, so I can just imagine if something doesn't work, there are so many places to look for improvement or error and stuff like that. And
yes.
PCA plus t-SNE, I just know that there's a lot of, especially t-SNE has a lot of hyperparameters, and you can just get lost in trying to optimize it, right? So I totally agree with you. You have to stick with what works. And
we had a postdoc, Claudia Battistine, who spent about a year parameterizing the pipeline. So the pipeline that's out there and that's freely available was generated for, or is optimized, I should say, for working with fruit flies. And they have their own sort of set of kinematics and behaviors and whatnot that they express, right? A fruit fly might a fruit fly might express 10 or 12 or 14 different discrete behaviors that, one framework will capture. But if you apply that to a rat, unless it's parameterized properly, you don't know what you're going to get. So it really, it sounds silly, but the fact is that, the power lies in the eyeball. And with the algorithm and the setup that we're using now, we're actually putting a paper together on it because we paired it with neural recordings in Cortex. We spent, me and the student, and it was great that we did this. We spent 2 days watching video output. There was labels that this thing generated for about 40 different behaviors. And we looked at the videos of the animals moving in 3D and looked at each and every single label and tried together, the two of us, with 2 sets of eyeballs to figure out and to put a name to the conglomeration of postural features that we thought the thing was capturing. So
you're sort of labeling them yourselves.
Yeah, exactly. That's the point. That's the whole point, is that if the algorithm works correctly, it's a totally unsupervised thing. It's doing is clustering variables through time. That's it. And if you want to apply a label to it, that has to come from the human to say, was the rat squirming? Was it squirming? Was it eating? Was it rearing? Was it turning? Was its head bobbling? So what you call the behavior is completely subjective. So when we were labeling them, we tried to avoid as much as possible like subjective interpretation, and just called it like walk slow, head tilt right, walk slow, head tilt right up.
Yeah.
And yeah.
That's fascinating. Do you guys then, do you label the correlation or the interdependencies between these movements? Because some movements are connected to each other. Some of them are because of the first one. Is there any sequence in these movements? Do you then also label the interdependencies between them?
So I can't answer that question yet. In a word, yes. So you can build what's called an ethogram. And I mean, here's the cool thing that's going on in behavioral neuroscience is that we're doing things that people have been doing since the 50s with the pen and paper. But now computers, I mean, because we've got better GPUs, we can do proper machine learning now without needing a Cray supercomputer or whatever, a quantum computer. And, we can use machine learning based approaches to produce unsupervised clustering of observed behavioral data and then give a name to it and study the statistics of it. And you can study the statistics, for example, by which the brain generates sequences of behaviors, and you can look at how that changes. So Bob Data is several years ahead of my group. They've been using their ARHMM, which they got working and published back in 2015, to study exactly this kind of thing that you're asking about. They're looking at, they call them behavioral syllables, just like you have like syllables in a word, right? You can decompose a word into its constituent syllables, just like you can deconstruct behavior into constituent syllables, like if the animal's rearing up or down, or if it's in the act of turning or twisting, right?
Right. Is that sort of kind of like the attention mechanisms in the sequence analysis? Because you have the new neural networks that kind of like the LNNs and LSTMs that try to analyze the long sequences, but then they always lose the, they have the memory attention problem where you always lose the where did you start, where you're going to end up, and then you can have these attention mechanisms that try to focus on kind of the word, the meaning of the word, or the meaning of the letter, like the small interdependencies within the structure of the word.
So let's see, I'm going to try to remember all the answers because like several things have occurred to me. So forgive me if I blank out or forget. You need methods to address, for example, so if you're talking about the complexity of behavior, right? Yeah. So I'm sitting here in the chair and I have a certain posture and I can tilt my head to the right and just do that and nothing else. But then I can tilt my head to the right because I'm going to look out the window, or I can tilt my head to the right because I'm going to talk on the phone, right? So there's pose features, and then there's the behaviors in which that pose is embedded. And then there are tasks in which behaviors can be embedded. So there's multiple levels of representation. It's like a hierarchy, right? Of like the physics of behavior, right? You can talk about kinematics. You can talk about how kinematics is organized. You can talk about behavioral organization. And there are different, there's no one method or another that is better or worse necessarily to ascribe brain function to one level or another or another. We have a set figured out. There will be a methods paper that we'll be sending out hopefully earlier next year, using information theoretics to try to find, which level is best explaining the spiking activity that we see of the neurons. The answer is in cortex, it's a sloppy mix, right? So you have representation of pose dynamics alone, and you have representation of the behavior in which the pose is embedded and so on. If we were at a different place, if we were a different lab, if I had many more machine learning people or much tighter collaborations with machine learning types, we would have probably gone down the restricted Boltzmann machine alley. That's what we were looking at first. So I said my former collaborator, Graham Taylor, and I was showing those videos of the people walking. We were going to use that exact same methodology and pipeline to try to understand rat behavior. But it never got off the ground. The ARHMM stuff from Bob Dada's lab came out, so we tried that instead of the RBMs because it was simpler. And then the ARHMM stuff didn't work, so then we have, we went this like a kind of hacky PCA TSNE route. If we had gone with the RBMs, the questions about like long short-term memory, vanishing gradients, there's a lot of terminology that has kind of like dropped out of my consciousness because we've moved away from RBMs and we've moved away from deep learning. But those are the types of problems that we would have been solving, exactly what you're asking about if we had stayed with the restricted Boltzmann machine, multi-layered deep learning kind of route.
Just kind of, sorry to interrupt, like, so how did you
I'll
then,
stop talking.
No, so how did you then circumvent that? So what is the kind of like, so what is the transition that you went from the older models that you don't actually have to take into account the memory loss and the oldest and the long sequences? How is it that you managed to kind of, what is it that makes the new system that you're working with that you don't need that to do any of these things anymore.
Well, it's not necessarily, I mean, so in the end, it doesn't matter if you're using a factored conditional restricted Boltzmann machine with certain long-term, short-term memory, whatnot, or if it's a shallow deep network, or it's whatever framework you're using that gives you data that you can look at with your eye and say, oh, it's working. It's working.
Oh, right. Yeah.
Got it. It's working. It's producing something that I believe. So that as you play the video and you're fact checking something, as you watch the video more and more, it makes your stress level go down because you're like, it works. As opposed to seeing a bunch of errors where you're like, shit, oh, this is not good. Like, gotta send it back to I send it back to Claudia. Yeah. So that for me, there's 100, 1,000 different ways to get to this sort of final goal of being able to temporally define when an animal engages in a certain behavior or not. What made us turn was just that the newer methods that were coming out were simpler. There's I have no other explanation. They were simpler, they were more adaptable. There are things that we could get to work, and they do work now, and they pass the eyeball test. So it ain't broke, and I'm not fixing it. And I'm not advocating for one method or another. Different labs have different approaches, and they tend to work beautifully, because somebody who's usually involved in machine learning and married to their network has worked very, very hard to get it to work very nicely. And what I've learned is that none of this comes cheaply. None of it comes quickly. It takes years and years and years of investment to get something to work. So once you have it working, don't break it, just keep using it.
Yep, that's the kind of general engineering thing that we're slightly learning more and more. If it works, don't touch it. Like if it works, then it's good. If it works, it ain't stupid, and don't touch it.
Exactly.
Yeah. Wow, that's but that was actually really fascinating what you were talking about with the unsupervised clustering. So it's kind of, you're basically taking all the data and then you're trying to figure out what sticks together, what things are actually correlated, like which things actually make sense together, and then you can kind of take away whatever way you can learn from that.
Yeah, the key is that you take something that varies through time. It doesn't matter if it's brain waves or increases or decreases in cerebral spinal fluid, or any variable that you have that's changing through time. If there are recurring dynamics, they can be clustered. And I just think it's an incredibly fascinating and powerful concept in the first place, to think about decomposing animal behavior into these physical elements that you can then study statistically. That opens up so many doors. And it's something that ethologists have been doing with the pen and paper since the 50s. The animal did this, the animal did that, the animal did this, the animal did that. And now we have machines that can do it for us. And all I can say is it's a very exciting time.
Oh, it has to be. I mean, to be able to quantify behavior, just any biological behavior, and actually put it down as a quantified number or an equation or a policy that you can alter, that would be amazing.
So cool.
It's so cool. It's fascinating stuff.
Yeah.
Let's see. We actually have a question from Jacob. Let's see. It's another question to Jonathan. Jonathan, are you ready?
I'm trying to think of who came up with deep squeaks. But no, that's another brilliant example of like using machine learning to. I'm sorry, I'll explain what deep squeak is.
Yes, what is deep squeak?
I cannot tell you about what's under the hood with deep squeak, but what I can say is that it's people had the idea. Okay, so you don't think of a rat or a mouse as vocal, but they are. We just can't hear them because they're outside of our range of hearing. So if you take a bat detector, right, which is literally that, it's an audio. It translates incredibly high frequency things that are out of our hearing range into an audible frequency. You can take a bat detector into our animal facility here, and if you put a male and a female rat next to each other, you'll hear that the male rat starts going woo 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 woo. You'll suddenly the animal will perk up and start singing. And I even had a graduate student or a couple of master students who were so enthusiastic and motivated. I wonder if I could find it.
I actually didn't know that rats were supersonic, just like bats.
Yeah, it's ultrasonic.
Yeah. Ultrasonic, yeah, that's right.
No, I don't know if I can find this or not. Oh, I wish I could. I have a video on my phone somewhere of my master's student who would play with the rat. And the rat, I mean, they're adorable animals. They are like pets. You just have to spend some time with them. And she had this thing going where she would stick her hand out, the rat would go back. She and the rat would kind of like fake each other out. And then she grabbed the rat and grabbed it and started tickling its belly. And she took with her, she had the bat detector out and you could hear the rat laughing as she was scratching the animal's belly. It was going woo, 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 woo. Every time she was stroking the belly, it was going woo, 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 woo. It's adorable. So, okay, great. That is, that's spectral, right? That's changes in sound through time. And there are different frequencies that are embedded in it. And rats basically have two different ranges on which they emit sound. One is around 50 kilohertz, which is a distress area, and the other one is around 20 kilohertz, which is a happy tone. I want to say it's like between 20 and 25 kilohertz, or 25, yeah, kilohertz. So they have distress and they have joy. But there's probably more to it than that. And the idea behind Deep Squeak is to basically take a deep learning algorithm and train it up on loads and loads and loads of emitted rat vocal emissions, and to cluster them and identify them, back to what the animal was doing when it made the sound, to try to, come up with like a rat lexicon, right? To decode the sound that the rat is making, based on what it was doing. So that's Deep Squeak, or at least as a consumer would describe it. So no, what I look into it, if given the time and resources, sure, I think it's very interesting. And it's a window into the animal's internal state, if the animal is stressed or if it's happy. So looking at the Rosetta Stone for rat language, really, you could call it that. Yeah, I mean, we could add sonics. We could add ultrasonics. We had a pellet reaching task before where mice were watching each other, where one mouse would be head fixed and watching another mouse grab food and eat it. We had a microphone. We wanted to catch any sort of like ultrasonic sounds that we could hear, but all we got was the animals crunching their food. But it is something that we thought about and tried to implement before. It's just difficult. And if, it depends on the task that the animal is engaged in as well, if it's going to emit sound or not. Right, but what we have in terms of tracking is the animals don't have a choice. They have a body, the body moves, we can track it. And in terms of what we want to expand and look at next, I think that the highest bang for buck thing that I could think of was facial tracking, where you just put two cameras, not 8, not 12, not 20, because some people do that. You just put two cameras over the face of the animal and look at where the eyes and whiskers are, because this is how they're sensing the world. And it opens the door to looking at sensory processing. It opens the door to studying the neural mechanisms of sensory engagement and the behavioral state of the animal to a degree, because the way that they move their whiskers tells you if they're investigating or if they're scared or if they're curious. So by using facial tracking, we don't have Deep Squeak, but we are looking at the faces of the animals we're starting to. It's a slow process, because every time you want to build a new tech and incorporate new tech, there's it never goes as fast as planned, but I mean, at least I can say it's on track. I would love to be able to start talking about the stuff this time next year. So knock on wood.
Right, yeah. Oh, that's fascinating. Stuff like that is always like just behavior analysis of just animals. Because I'm just, well, while you were saying that, I would keep thinking that, as you said, when like you have your eye that you can tell what is actually going on. And then like I have cats, right? And then I know the behavior of my cats, but only I know the behavior of my cats. When he comes in, I can tell the way they move, the way they are. And then can just imagine if we have this algorithm that can analyze animals or pets and then actually tell you what is it that your animal wants to do. How does it feel? Does it feel bad? Does it feel sick? Does it feel sad? And these are exactly. And these are like kind of a pet analysis. That would be a fascinating thing to do.
Right. So, I mean, the idea that, I mean, ultimately the idea is to use machine learning algorithms to enable us to do the same thing with our animals in the lab. And when you realize that, it opens up so many questions that you couldn't access before. And it makes me a little bit crazy that the field has gone on as long as it has, basically neglecting behavior.
Yeah, it's very weird, actually very odd. Once you said that, I've never thought about it. It's very little behavior analysis. There's so much AI analysis and forecasting and any form of, but except for there's very little I've heard research in behavior analysis.
Well, I can say at least it's happened and it's taking off.
Yeah,
more and more labs are doing it, and there's been a lot of advances in the invertebrate community, people studying worms and flies and that kind of thing. So the worm, you think, why is a worm interesting? Actually, if you have the right tools, the worm is like the most fascinating thing you could possibly study, because you pick the right worm, it's got 300 neurons. You can know actually
doesn't it have like a lot of bodies, and you can cut it in half and it'll actually be divided into two separate animals, two separate entities?
But the interesting thing about using these lower organisms is that they have fewer neurons, right? So you can build a more complete model of their brain and how it's working, and you can understand maybe more deeply how the brain of that animal is actually generating behavior that you can measure. I think with the rats and mice, we're just scratching the surface, because these animals have 100 million neurons.
And how much can we analyze at this moment?
In terms of behavior or neurons?
Neurons.
Neurons, tens of thousands of cells at a time, max.
Right.
Like individual neurons.
Yeah.
Tens of thousands.
Right.
It's happening. But in those, that only works with very special microscopes. Right.
Well, what about BCIs? Actually, that are intrusive BCIs, the ones that are actually installed inside the.
Yes. Well, so if you want to get like a jillion megacells, you usually have to lock the animal's head so that an objective microscope can fit over the animal's head. That's connected to a scanner of some sort and a very, very powerful laser to excite. I don't want to get too deep into the methods, but it usually comes at the cost of restricting the animal's behavior. What's cool is that better tools are emerging that you can use in freely moving animals. And now it's possible to have, let's say, a few hundred or maybe even a few thousand cells at a time in a freely moving animal. And then if you compare that with powerful behavioral analysis, then you're cooking with gas. There's a million questions you can answer, or a million questions you can ask.
Right. I think we're starting to run out of time. If there's any questions or any, let's go ahead.
I heard about a company that was making surveillance and pattern of behavioral pattern matching for cows. So basically then seeking if it was sick, lame, or whatever. And what they did was actually to apply like 4K cameras and stuff like this, because then you can really start what do you call it, recognizing them, because in 4K you can see even differences when they are what is called monochromatic, when they are all black, let's say, right?
Oh yeah.
So you can see differences in fur and whatnot. And there was also a study on pigs when they were drinking, so basically coming up and then face recognition on those. And there's a lot of that. So basically that technology just getting cheaper and cheaper also allows to right. So you don't have to put in trackers and whatnot, but as you said, the grabbing of the rat and putting something fascinating.
Yeah. So if you want to just track an individual and just put a dot on the individual and not like anything about like the 3D kinematics or anything, that's easy and that's very cheap. And I can only imagine maybe it's being used in farming or livestock if you want to keep track of animals.
Yeah, I was just
thinking colonies or
maybe cooperation, right? Because some people are already done very specific systems on, let's say, cows, right? And then see if one could borrow some of the technology, right? Or...
Absolutely. Yeah, no, I mean, you said the word. I mean, it's getting cheaper and cheaper. It's getting, I mean, it's becoming more, pervasive as time goes on. There's more, I can't list them all, but I mean, there's certainly more and more applications. I reviewed a paper, a couple of months ago about, it was about tracking chickens in a coop. And I mean, they weren't using any kind of like monstrously big framework to do it. And the question was quite simple, but it was a very robust thing. And it was very eye-opening to me about how, using these types of tracking methods that you're mentioning here are going to benefit the agriculture and livestock industries, because you can see if an animal is sick, for example, without having to go in and check on them. You can have an algorithm tell you, so-and-so just gave birth.
Because there was actually, this company brought up a very interesting thing. When you have cows, they are herbivores and we are carnivores, right? So basically they just, if they are sick, they behave differently when there is a farmer around, because they're just like, okay, I'm not sick, and there's all this aspect of earmarking and whatnot. So it's let's not harm them, right? So, so as you said, a couple of examples of restricting animals, and this is just opening a whole new segment of analysis, right? Where they just thrive and do whatever they used to do.
It is,
That actually makes total sense when you think about it, because if you're trying to have behavior analysis of any entity, and if then you confine that entity to a specific area, then it's not behavior, it's not natural behavior, and it's just somebody in a cage. Like how is that a behavior analysis? Then you basically, it's a deterministic behavior analysis where it doesn't really give you all of the answers.
Yeah, and that's what the tools are allowing us to break from, is this, as you said, deterministic things that are preconceived, a priori defined by an experimenter or someone to say, yeah, the animal wants to do this, the animal's going to do this. And I think, it's kind of an ass backwards way to understand behavior, like to force it to happen and then backtrack and say, how did the brain make that behavior happen that I told the animal to produce?
Yeah. I think it's not really behavior analysis, because you're not actually checking what the animal is doing. You're telling the animal to do it, and then you say, yes, he did it, and then you're satisfied.
Yeah. Well, it's not, I mean, it's not a useless approach. It's not a useless approach at all. It just restricts the kinds of questions that you can ask. And
but then again, it's kind of like you need the back end data to understand. So when you make an animal do something, then you read the neurons and create this kind of neural pathway of this is how the animal behaves when this is happening. And then you do this unsupervised clustering, and then you try to match them up, and then you say like, oh, this cluster actually relates to them trying to run or something, right? So this kind of gives you a little correlation between some of the unsupervised clustering and something that was actually deterministics, and then analyzed, and then you know for a fact that this cluster is whatever it means.
Yeah, I think that the key, and another key word that you said is deterministic, right? That in order to get that kind of control and precision, you used to have to determine, how's the animal going to be positioned? What's the animal going to do? What decision are you going to allow the animal to make? And now, again, with machine vision and all the things from my talk, we can use tools to ask questions with freely moving animals that are producing their own behavior at a level of precision that kind of starts to approximate what happens in those predetermined scenarios, right, where the animal's restricted. So it's just really exciting, because the brain evolved to generate adaptive behavior. Yeah. Now we can actually study adaptive behavior, like the way the brain intended it.
Yeah.
not the way the scientist intended it.
Yeah. Wow, the future is just getting more crazier and crazier. It's getting so cool. So much stuff coming out right now. All right, it's 5:30. I do believe that we are wrapping up. For those that are interested, there is some networking in GatherTown. The link is on the Oslo Met AI. I will post in chat just to be sure, so that everybody has that. And there we go. For some reason, oh, that was too. Here we go, everyone. There we go. So there's a meeting, and please also visit us. The link is on the AI meetup page of neuralmet.no or oslomet.no, which is me and Jürgen's university, and neuralmet.no is our student organization that we are trying to kind of introduce and get people more excited about AI. And I would like to thank everybody for joining, and thank you to all the participants. It was really lovely and extremely interesting conversation. Thank you so much, everybody, and have a nice evening.
Thanks. I will not be able to stay for the meetup. I have another engagement I have to go to that starts at 6, so I'll have to go.
That's fine.
Thanks again.
Okay. Thank you so much for an interesting thing. Bye-bye.
Good night. Thank you, everyone.
Thank you. Right. Stephanie? Now there we go. And then